Hey guys, welcome back to the channel. So in this video we'll talk about functional, comparative and structural genomics. So let's get started. So talking about functional genomics, so why do we need and what are the methods for extracting the needed portions for functional genomics? So why do we need is we get to understand the gene expression, we get to know the gene regulation, we also understand about gene-wide mutagenesis. And how do we do so? There are different methods for doing so. So you can see the number of methods listed here on your screen, which is data mining, SAGE, microarray analysis, subtractive cDNA libraries, yeast to hybrids, transgenes, transposon targeting, RNAi, mRNA. So there are different methods for extracting the known features for functional genomics. So what is its utility? So to understand the utility of functional genomics, so we need to get through some of these points which are given on your screen, such as the ultimate goal is to define the function of each gene in a genome. So the ultimate goal is to define all the functions of each gene in a genome. So the functions can be categorized at different levels, from biochemical to cellular to organisms. So the functions of these genes in a genome are categorized different ways, from it can be biochemical, cellular to organisms. So depending on the levels, it is segregated. Also we have homologous genes in different organisms often have similar functions. So the name homologous suggests that there is a similarity. So the homologous genes in different organisms often have similar functions, but there are many exceptions as well. So there are different techniques under functional genomics. So as we just talked about in the first slide, there are different techniques for extracting gene expression, regulation and its mutagenesis. So we'll get deep into those methods right now. So some of the techniques are subtracted cDNA libraries, we have differential display, we have representational difference analysis, we have suppression, subtractive hybridization, we have cDNA microarrays, we have serial analysis of gene expression, which we call as SAGE, we have 2D gel electrophoresis, we have RNA interference or RNA silencing. So there are these number of techniques that are involved under functional genomics to extract the expression of a gene. So moving with that, so why do we need functional genomics? So as you can see, the different organism name, different columns are mentioned with their organism's name, such as E. coli, yeast and all of them. You have the gene number as well depending on the organisms, and you have the percentage of genes with inferred functions. So from this technique, from this functional genomics technique, so we have got the percentage or the number of genes that have a specific assigned function. So the number of genes which have functions are calculated from the total number of genes that are present in an organism. So from there a percent has been calculated by the total number of genes present in an organisms, so that we get to know how many genes are important, what is its function and how it is regulated. So these are the number of gene percentage. So this is 60%, we have 40%. So these are the percentages given with inferred functions. Also we have completion date of genome. So these E. coli genome was completed in 1997, and along ways we have other organisms with their dates as well. So talking about the next part, which is comparative genomics. So talking about comparative genomics, which is a bit different from the functional genomics. So you can see circles of gene synthetic relationships. So this is the picture in front of you, alright. So it means information gained in one organism can have applications in other. Alright. So this is a very important point in comparative genomics. So information gained in one organism or information inhibited by one organism can have applications in other organisms as well. Even distantly related organisms are applicable to this. So this is a circle of gene synthetic relationships. So there are different you can see oats, triticale, maize, And different food items you can see made in a circle or through the gene number and everything. In a form of a genome they are created, and through this they are trying to find the mutual relationships between these items, so that the one of the functions inhibited by one of the food items can be beneficial to the other as well. So this is how they are trying to find the relationship between 2, 3, 4 and different organisms or different food items you can say, or different cereals, spices you can say, so that it becomes a common point for all of them. So moving more to understand more about this, so analyzing DNA sequence patterns of different organisms side by side to identify genes and determine functions. So you can see two DNA sequences you can consider. There are two DNA sequences present here of two different organisms or of the same organisms. To compare the two different DNA sequences and to check their similarity and their differences, and to identify some of the common genes and determine functions. Alright, also looking for similar genes in different species to determine possible relationships and genomic variation. So as I said, if there are two different DNA sequences present here, so we try to find out the similarities and the differences between the two sequences, and we try to find out those genes which are similar and the genes which are dissimilar, and try to find out the relationships, the mutual relationships between the two DNA sequences and the variations they carry with their dissimilarities. So what are some questions that comparative genomics can address? So these are some of the questions that the comparative genomics can solve actually. So how has organism evolved? So basically it helps us to find out the root cause or the birth of an organism, so where the organism is developed or evolved from. So we can find out the root cause of that particular organism. What differentiates species? So we can understand the differentiates or the differential identity or something that differentiates these two species with the gene similarity or dissimilarity in the relationship or something. Also, which non-coding regions are important? So definitely it is very important to understand that if there are two DNA sequences, we need to pay heed to both the coding and the non-coding sequence. So people generally look for coding sequence, but at the same time non-coding sequences are equally important to find out the dissimilarities and the relationships between two different organisms or of the same organism, but there must have some similarities or dissimilarities. So with the help of that we can find the some of the non-coding regions and figure out that genomic variation. Also, which genes are required for organisms to survive in a certain environment. So from there we can also find out the difference in between two or the similarities in between the two organisms, and we can figure out which genes are very much essential for the organism to survive in a particular environment, and which genes are not very much important for an organism for survival. So these are some of the questions that comparative genomics can solve. Moving with that, so talking about the last part, which is the structural genomics. So talking about the structural genomics, so it characterizes the physical nature of the whole genome. So it helps us to find out or helps us to characterize the entire genome of an organism. So it compares different genomes of an organism or a species, and from there it characterizes those genomes depending on their nature. Also it includes genetic mapping, physical mapping, we have sequencing of entire genomes. So the sequencing part, the mapping part is involved in the structural genomics. So the entire genome of an organism is taken at once, and not the DNA sequence of an organisms are taken. So the entire genome consisting of all the genes of an organism are taken here. So this was the picture I want to show you. So this is the overview of the general approaches of structural genomics. So you can see here sequence organization, we have assigning loci to specific chromosomes. So different locis are assigned. Also high resolution chromosome maps are done, or mapping is done. Physical mapping of genomes, genome sequencing, as well as using genome maps in genetic analysis. So there are different functions that are involved here, taking genomes or the entire genome of an organism at once and thereby assigning them and mapping them according to their similarity and their gene uh, functionality. So let's just keep this video till here. Hope you enjoyed this and thanks for watching.